Hello everyone again. I welcome you all to the Introduction to Developmental Biology Week 11 PMRF-NPTEL lecture. As if you have followed the previous videos, you already know that the PMRF-NPTEL lectures are designed so that a PMRF fellow such as myself can sit down and interactively solve various assignment problems of that week's PMRF course. In our case, it is Introduction to Developmental Biology. And by solving the problems along with the students who are taking the course, the students get a better idea of how exactly to approach the week's assignment problems and have a better understanding of how to tackle the course material and also develop a deeper understanding of the course material as well. So this is the week 11 lecture. So let us get started. First, I would like to introduce myself. My name is Anuvab Chatterjee. I am a PMRF Fellow and PhD Scholar under the supervision of Professor Jonaki Sen at the Brain Developmental Biology Lab, Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. In this week's lecture, we had specifically gone through the early mammalian development, looking closely at placental development, and had a brief introduction towards evolutionary developmental evolutionary developmental biology or EvoDevo. So those are what the questions are for this week's assignment as well, and those are what we will be going through today. So let us get started. This is the first question that we will tackle today. This is the question is embryonic mesoderm arises from which of the following type of cells? So if you remember correctly that various tissues found from the embryo have various lineages. We had already seen how the two layers epiblast and trophoblast divide, which then gives rise to the endoderm, mesoderm and ectoderm. So the particular question asks us what is the lineage of the embryonic mesoderm? Is it from the hypoblast cells, the primitive groove, the epiblast cells, or the embryonic ectoderm cells? So let us. So will anyone try and answer this? This is a round type of question. So only one option is correct. Embryonic mesoderm arises from which of the following cell types? Hypoblast, primitive groove, epiblast, and embryonic ectoderm. Okay. So let us get into the solution slide and try to answer this. So this is a schematic that shows us the lineages of various cells coming from the right, starting from the high blastocyst and ending with the various uh, embryonic tissues such as embryonic ectoderm, embryonic endoderm, embryonic mesoderm, as well as extraembryonic tissues such as extraembryonic mesoderm and the syncytious trophoblast, which will uh, give rise to the embryonic part of the placenta, that is the chorion. So here from the diagram itself, you can see that the embryonic mesoderm is actually from the epiblast lineage. The hypoblast gives rise hypoblast as well as the epiblast both together give rise to the extraembryonic mesoderm, and more or less since the embryonic ectoderm, endoderm, and mesoderm contribute directly to the embryo, they would obviously come from the epiblast. So that is the essential difference between epiblast and trophoblast. Trophoblast cells are placental cells that will give rise to the connection between the embryo and the mother, whereas the epiblast cells gives rise to the actual embryo. So anything that has embryonic in its name will literally arise from the epiblast. So thus the answer to the question is embryonic mesoderm cells arise from the epiblast. So does anyone have anything to ask regarding this? Sure. So we move on to the next one. The next question is, which of the following are correct about the node? So for the context, node is a particular structure that forms at the end of the primitive streak. So if you remember correctly, during embryonic development, first there is the formation of a bilaminar disc, which is the epiblast and the trophoblast. So after that, after the formation of the disc, epiblast and hypoblast. After the formation of the disc, we see that a primitive streak occurs, via which cells start migrating into the hypoblast from the epiblast. At the posterior end of the primitive streak, there is a structure that forms, which is known as the node. This is an embryonic structure, and it secretes various molecules which help in the development of the embryo and establishment of the anterior posterior plan. As we had seen in Drosophila development, it is not similar to Drosophila development in such a way that in Drosophila development, maternal effect genes such as caudal or becoid, as we had seen, play a major role in establishment of the anterior posterior patterning of the embryo. But here in mammalian development, mammalian embryonic structures play a vital role, such as the node, and we will also look into another structure which is known as the AVE, play a major role in determining the anterior posterior pattern. So node is at the posterior end of the primitive streak, and it secretes various molecules and helps in the patterning of the embryo. So which of the following are correct regarding the node? We have four options. This is node is at the anterior end of the primitive groove. Node is a structure formed in the hypoblast layer. Node is a signaling center involved in axis formation, and node secretes caudin. Caudin is a molecule which has various roles in development. Will look into caudin momentarily in the next slides. So this particular question has more than one correct because it's a box type of question. So will anyone try and answer this? Okay. So let us go ahead with the particular question and try to answer this. So this is what the structure exactly looks like. So in the first part of the picture here we have GFP labeled cells, and we are trying to trace the direction in which the cells are actually migrating. This is known as the stereogram. So this the A part is a stereogram of gastrulating chick embryos showing the relationship of the primitive streak, the migrating cells, the hypoblast and epiblast of the blastoderm. So the cells on top are the epiblast cells, the cells here are the hypoblast cells, we can see right here, and we can see that cells are migrating into the primitive streak. This is the primitive streak right here, and with the this structure being the Hensen's node, or the node as we were calling it. The lower layer becomes a mosaic of hypoblast and endodermal cells, the ones that are migrating inside. The hypoblast cells eventually sort out to form a layer beneath the endoderm and contribute to the yolk sac. Above each region, the stereograph or micrograph showing the tracks of the GFP label cells at the position in the primitive streak. So we are able to visualize how exactly the cells look as they are migrating through the layer. So this broad line is the primitive streak, and these are the cells that are going into the plane, and that is into the via the primitive streak they are going into the embryo. Cells are migrating through the Hensen's root, travel anteriorly to form the precaudal plate and the notochord. Those migrating through the next anterior region of the streak travel laterally but converge near the middle to make the notochord and the somite. The somite is the structure which gives rise to the muscles and the bones of our spinal cord, the vertebrate column, and from those from the middle of the streak form the intermediate mesoderm and the lateral mesoderm. So these are the various structures that are formed by the migrating cells, and here we can appreciate that the node is in this position of the primitive streak. So this is what the location of the node determines for us. Next, let us look into what exactly is the contribution of the molecules that are secreted by the node. So here we have a three-dimensional model of the developing mouse embryo, and here we the mouse embryo develops as a cup-like structure with these two embryonic structures which I said were essential: the node, which is right here at the bottom, and the anterior visceral endoderm, which is the red yellowish structure which is shown right here. Both of these are important signaling centers, and for the development of the mouse embryo or any embryo or mammalian embryo, the AVE produces lefty 1 and cerebrus, both antagonists of nodal. This restricts the formation of primitive streak and node to the posterior. In order to maintain the correct positioning of the primitive streak so that it does not extend towards the much towards the posterior, the AVE releases these signaling molecules, lefty 1 and cerebrus, which prevent the progression of the primitive streak towards the posterior end. Node in turn produces cordon. Notochord formed from the node produces noggin. So as the embryo develops further, the node and the AVE will be linked by the notochord, which is another embryonic structure, which produces another important signaling molecule, noggin. Both cordon and noggin are BMP antagonists. That is, they prevent BMP signaling in the region that they are secreted, and these to promote the formation of anterior structures. So here we can see a cross-sectional view of the node, and in the yellow we can in the white we can see the cells that are migrating into the embryo. So this is the caption to the particular image: the axis and notochord formation in mouse embryo. In a 7-day mouse embryo, the dorsal surface of the epiblast or the embryonic ectoderm is in contact with the amniotic cavity. The ventral surface of the epiblast contains a newly formed mesoderm. In this cup-like arrangement, the endoderm covers the surface of the embryo. So we can see the yellow layer, that is the endoderm covering the surface of the embryo. The node is at the bottom of the cup and it has generated the chorda mesoderm. The two signaling centers, the node and the AVE, are located on the opposite side of the cup, and eventually the notochord will link them, as I said. By embryonic day 8, the AVE lines the foregut, and the precaudal mesoderm is in contact with the forebrain ectoderm. The node is now further caudal, largely as a result of the rapid growth of the anterior portion of the embryo, and the cells in the midline of the epiblast migrate through the primitive streak, which are shown here in white arrows. So the two important points that we can get from these slides is, first, that there are two important signaling centers which help in the anterior posterior patterning of the embryo, which are the AV and the node respectively. The node is at the anterior end of the primitive streak. The AV releases leftian cerebrus, which are antagonists of nodal and prevent the progression of the primitive streaks posteriorly. And the node releases cordin, and the notochord that joins the node and the AVE releases another important signaling molecule, which is noggin. Both of them are BMP antagonists and help in the formation of anterior structures. So, going back to our question itself, we can conclude that node is a signaling center involved in axis formation, and node secretes cordin are the two correct answers. So that was a long discussion. I would like anyone, if anyone has any questions regarding this, they can just go ahead and like post it in the chat or try asking any questions regarding the discussion. And for node in the anterior end of the primitive groove is not correct, because primitive groove is not the primitive streak. It's another structure where the cells start migrating first. If it was primitive streak, then the first option would have been correct as well. So if anyone does not have any questions, we can move right on ahead. This is the next question that we are looking into. The question is, what is the role of AVE in the early mammalian development? The options are: it produces antagonists of BMP signaling, it produces lefty and cerebrus, can form the entire body structure by itself, and it prevents formation of primitive streak and node towards the posterior. This is a box type of question, so there are more than one option that are correct. So if anyone will try and answer this, like even guess the answers to the question. Yeah. So let's just go into this slide again and try and answer this. So this time around, we are looking at the antevisceral endoderm in detail. And as we had already discussed, EVE produces leptin cerebrus, which is antagonist of nodal, and it prevents the progression of the primitive streaks towards the anterior as well as helps in the formation of anterior structure. Hello. So we are discussing this particular question, which is what is the role of AVE in early mammalian development? And the options were it produces antagonists of BMP signaling, it produces the signaling molecules lefty and cerebrus, it can form the entire body structure by itself, and it prevents the formation of the preventive streak and the node towards the posterior. So this is a more than one option, correct type of question. So would anyone like to try and answer this? Okay. So let us go into the solution slide and try to answer this again. So as we had just discussed, there are two important signaling centers in the developing embryo. Those are the AVE, the anterior visceral endoderm, and the node. The AVE, as we had just discussed the node, we now discuss in detail about the AVE itself. The AVE is the signaling center that releases the molecules LEFT1 and cerebrus, which is antagonists of nodal. These release of these two signaling molecules by the AVE prevents the posterior shifting of the primitive streak and marks the boundary of it. This in turn promotes the formation of anterior structures and help define the positions in which the gastrulation will occur, that is where the cells can actually move into the embryo, as we can see in this white arrow right here. So that so node marks the boundary of where exactly the cell migration will stop, and AVE determines that via the release of lefty1 and cerebrus. There is also one another point that we have to keep in mind is that via various experiments it was determined that if the AVE is disturbed, it is seen that the body or the structures of the body can still develop via the action of the node. The release of caudin and noggin by the node and notochord respectively can give rise to the almost give rise to the entire body structure, but the AVE alone cannot do that. So returning back to a particular question, what is the role of AVE in early mammalian development? The answer is it produces lefty1 and cerebrus signaling molecules, and it prevents the formation of primitive streak and node towards the posterior. It does not produce antagonists of BMP signaling, but instead it produces antagonists of nodal, and it cannot form the entire body structure by itself. So does anyone have anything to add to this? Okay, so we move on ahead and look at the next question, which is blood vessels of the chorion are made up of which of the following types of cells? The four options are extraembryonic mesoderm and trophoblast cells, just extraembryonic mesoderm, cells of the maternal blood vessels, and syncytiotrophoblast cells. So this is only one type of correct answer because it is a round type of question. So would anyone try and answer this? Okay. So let us look at the solution slide and try to look into the problem. So now we have shifted from gastrulation to, and we are now talking about development of the placenta itself, that is the connection between the embryo or the fetus and the mother. This is done via the trophoblast cells. The trophoblast cells give rise to two lineages. One is the cytotrophoblasts and syncytiotrophoblasts. The cytotrophoblasts are responsible for the initial adherence of the embryo and the mother's endometrium layer. Next, the syncytiotrophoblasts help in further burrowing of the embryo into the mother's uterine wall and starts the dissolves the endometrium wall and lays the foundation for formation of further blood vessels. The blood vessels themselves are essentially formed by the extraembryonic mesoderm, which joins the trophoblastic extensions and gives rise to the blood vessels. The extraembryonic mesoderm ultimately forms the vessels of the umbilical cord. So the extraembryonic mesoderm is essentially responsible for the formation of the umbilical cord itself. And trophoblast cells, tissue and blood vessel containing mesoderm is known as the chorion. So we had the picture here. Yes, we have the picture right here. So as you can see right here, this structure, this structure which ends up into blood vessels which look something like this coming from the fetus itself, is the embryonic part of the placenta, which is known as the chorion. And the blood vessels coming from the mother, and the structure, endometrium structure right here, is the mother's part of the placenta, which is known as the decidua. As you can see right here, that we have two arteries forming from the fetus and one vein, and these are not directly connected to the mother's circulation in any way. These are what happens instead is that the mother's circulatory system and the fetus's circulatory system act via diffusion. So the oxygenated blood enters directly into this trophoblastic cavity, in which diffusion occurs and no direct contact between the blood vessels is there. The embryo takes up various nutrients directly from this trophoblastic, the mother's placental decidual cells via diffusion, and releases its waste product directly into this space instead of directly being connected to the mother's circulation. So this is what happens. This is how exactly the placenta works, and in the entirety of the blood vessels is formed from the extraembryonic mesoderm, whereas these structures, the chorionic villi structures, as or this lining, the lining is entirely formed from the trophoblastic cells, or especially the syncytio trophoblastic cells. And here we have a diagram showing the human embryo and placenta after 50 days of gestation, and this is how it looks in the live embryo. And this particular structure is the big structure in the end, with a large number of networks, is the placenta. So going back to a particular question, blood vessels of the chorion are especially made up of the extraembryonic mesoderm. Does anyone have anything to add to this? Okay. So we move right on ahead, and the next question is, arrange the following events in the appropriate order for the formation of the placenta during early mammalian development. So we have 5 points, and we have to arrange them in such a order that in the correct chronological order, so that the placenta develops. So the 5 options that we have here are: number 1, the extraembryonic mesoderm forms the blood vessels of the umbilical cord. Diffusion of the nutrients and waste products initiate between the fetus and the mother. Cytotrophoblasts provide initial adherence to the endometrium via adhesion molecules. Maternal vascular tissue is replaced by the trophoblast cells, and syncytio trophoblasts further progress the embryo into the uterine wall. We have 4 options, and obviously only one is correct because the numbers are different in every one of them. Okay, so Oindrila says the option is 35412, that is option number 2. Does anyone else want to try and answer this? Okay, so let us look into solution slide and then go back to this particular answer. So we had already gone through this slide, and this is on this slide the question is based. So what is the order of the things that happen? First is the initial adherence to the endometrium through adhesion molecules, that is done by the cytotrophoblasts. Once the adhesion is done, next the syncytio trophoblast further progress the embryo into the uterine wall, that is the second point. The third is the maternal vascular tissue is displaced by these trophoblast cells. The fourth is the extraembryonic mesoderm giving rise to the blood vessels and the umbilical cord, and the fifth is the exchange of the nutrients and waste products. These are the correct order. cytotrophoblasts for giving rise to the adhesion, syncytial trophoblasts help in further progression, syncytial trophoblast cells replacing the mother's maternal vascular tissue, extraembryonic mesoderm forming the blood vessels and the umbilical cord, and finally the placenta starts working and there is exchange of nutrients between the fetus and the mother. The correct option is 3. The cytotrophoblasts provide initial adherence. After that, 5. Syncytial trophoblasts further progress the embryo into the uterine wall. Then 4. Maternal vascular tissue is replaced. Then 1. Extraembryonic mesoderm forms the umbilical cord, and 2. Diffusion of nutrients on waste products. So the correct option is number 2, as Oindrila suggested. So does anyone have anything to add to this particular question? If not, we can go right ahead and look at the next question. Absence of the syncytial trophoblast cells in the early embryo prevent the formation of which of the following? So if the syncytial trophoblast cells were absent from the fetus, let's say, then which of the following structures will not form? Will it prevent the formation of the decidua? Will it prevent the formation of the chorion? Will it prevent the progression of the embryo into the uterine wall, or it prevents the adherence of the embryo to the uterine wall? This is more than one type of answer because it's a box type of question. So Oindrila says it is 2 and 3, which is it will prevent the formation of the chorion and prevents the progression of the embryo into the uterine wall. Anyone else wants to try and answer this? Okay. So again we go back to the same question, same slide. So the syncytial trophoblast cells help in, as I said, for the progression of the embryo into the uterine wall. So that if syncytial trophoblast cells are not there, this is the first thing that will get inhibited. This embryo will not progress into the uterine wall, and since the embryonic portion, then the embryonic portion of the placenta will obviously not form, which is known as the chorion, and thus it will also prevent the formation of the chorion itself. The correct answer is 2 and 3, just as Oindrila suggested. So does anyone else wants to try and answer add something to this question? Okay. So we move on to the next one. So next we look into further aspects of mammalian development and look at the formation of the embryo itself. Now, so the question is, during human embryo development, what will be the consequence or consequences if the cells separate at a two-cell stage? So if the embryo itself divides into two at the two-cell stage, then what will happen? The options are: embryonic development will abort, this will result in the formation of monozygotic twins, two embryos will develop in a common amnion and chorion, and two embryos will develop in two separate amnions and chorions. So this is more than one option, type of correct type of question. So will anyone try and answer this? Okay. So Oindrila has suggested, go back to the tab and look at it. Chatterjee. So Oindrila suggested the correct option is 2 and 4, which is each cell gives rise to their own ICM and trophoblast. So 2 and 4 is correct according to Oindrila. Anyone else wants to try and answer this? This. So 2 and 4, Oindrila says that options are this will result in the formation of monozygotic twins, and the two embryos will develop in two separate amnions and chorions. So let us look into the particular solution slide and try and understand this. So this is the diagram which shows us what will happen if the embryo divides into two at various stages, especially looking at the two-cell embryo stage. The options that the various things that can occur is that first, the zygote in the first division divides into two, and since each the zygote is still totipotent, it will give rise to both the trophoblast layer and the ICM. So, if the embryo divides into two at the two cell stage, we have in two entirely separate trophoblasts and ICM layers. And since the trophoblast gives rise to the chorion and the ICM gives rise to the amnion and the embryo, we have two completely separate chorion and ICM combinations which look like this. So we have two chorions for each of these trophoblast cells and two amnion as well. So if the zygote divides into two at the two cell stage, we have two chorions and two amnions. But if it divides such that only the ICM separates into two and we still have one trophoblastic layer, since trophoblast will give rise to the chorion, we have one chorion and two amnions as seen right here. One chorion and two amnions. And finally, if the separation occurs even later, so that after the separation, after the ICM has separated out, then the cells divide into two. So we see that the this particular zygote will give rise to only one amniotic cavity, because the amniotic cavity is determined by the ICM. So after a particular stage, once the amnion has started to develop, if after that the embryo divides into two, we have two embryos developing in only one amnion and one chorion, obviously, because there is only one trophoblastic layer. So this is the particular caption of the particular schematic. This shows the formation of human monozygotic twins with respect to the extra embryonic membranes. If the splitting occurs before the formation of the trophoblasts, each of the twin has its own chorion and its own amnion. If splitting occurs after trophoblast formation but before amnion formation, resulting in twins having individual amniotic sacs but sharing one chorion, and splitting even after the formation of the amnion leads to the twins in one amniotic sac and a single chorion. So, a correct answer to our question is, if the cells separated at two cell stage, the cells will result in the formation of monozygotic twins, and the two embryos will have two separate amnions and chorions. So this is also as Oentrela says suggested, this was the correct answer. So does anyone want to add anything to this? Okay. So we move on to the next question, and now we are looking at further looking at development, and now we are looking at various signaling molecules and genes which play a role in development, early mammalian development. So the first thing that we will look into is retinoic acid. What is the role of retinoic acid or RA during mammalian anterior posterior embryonic patterning? The options are: it will inhibit Hox gene expression, it will activate the gene CDX1, it negatively regulates FGF8 signaling, it activates Wnt3A signaling. This is only one type of correct option because it is a round type of answer. So if anyone wants to try and answer this. Oendra says the option is number 2, that is activation of CDX1. Anyone else wants to try and answer this? Okay, so let us look into the particular slide and try and answering this. So in order to understand important aspect of early embryo development, we have to understand what exactly is known as a organizer. A organizer, if we had looked into amphibian development as well, but we would have understood better about what an organizer is. But essentially what organizers do is that this mark a specific position in the embryo which will give rise to the anterior posterior pattern. So this particular organizer will is the major determinant factor in which determines how the patterning of the embryo will look like. So how does the organizer do it? The organizer does this by a very simple method. One side of the organizer, or one the what portion, say the posterior portion, it activates particular signaling molecules and inhibits them in the post anterior section. So as we can see right here, in the posterior sections of the organizer, Wnt's, BMP's, or FGF signaling are active, and in the anterior they are all inhibited via BMP and Wnt antagonists, and hence we have asymmetry that develops, and this is how the entire anterior posterior planning occurs. This so this can act via various molecules, and one of them includes retinoic acid that we will be looking into right now. So retinoic acid, Wnt signaling, and FGF8 signaling, what do they exactly do? These signaling molecules, after being determined to be expressed in a particular pattern by the organizer, give rise to the activation of a certain set of transcription factors which are known as CDXs. CDX transcription factors in turn activate the Hox genes, and as we have seen, the Hox genes are very important in the anterior posterior planning, and these are essentially the body planning or the body patterning genes. And as you can see directly from this image as well as the C image, if a particular signaling is present at a particular location, only that CDX will be expressed. That CDX expressing means the Hox gene downstream to that transcription factor will only be expressed in that region, and only the structures that are formed by those Hox genes will be developed in that region in the developing embryo. So essentially, the combination of the signaling molecules in the particular embryo or the particular region of the embryo gives rise to the anterior posterior pattern. So what retinoic acid does, it activates the transcription factor CDX1, and this in turn gives rise to the activation of various Hox gene downstream to it. Similarly, we have other signaling molecules such as Wnt's and BMPs, which also play a similar role. So here we have the caption of the particular image as well. This particular image shows us the concentration gradients of BMPs, Wnt's, and FGS in a late gastrula mouse embryo. Although it is shown as a flattened disc, but the mouse embryo looks like a cup-like structure as we had seen in the previous image. And here in this B portion we can see in situ hybridization image of FGF8, which shows us the concentration gradient of FGF8 as well, being highest in the red portion and lowest in the green, which is according to the diagram shown right here. At the organizer, the FGF signaling is the lowest, and in the posterior region it is the highest. So this is how the patterning occurs, and in reference to our particular question only, what is the role of retinoic acid during mammalian anterior posterior embryonic development? It directly helps in the activation of CDX1 and thus helps in the expression of Hox genes. Does anyone have anything to add to this? If not, we can go right ahead into the next question. So we are looking at another aspect of retinoic acid during development. This is at a bit later stage where the embryo has developed to a particular point, and the question is, presence of excess retinoic acid during early development gives rise to which of the following defects in the embryo? The options are: it will cause a shift in anterior structures towards the posterior, it will cause a shift in posterior structures towards the anterior, it will inhibit the formation of the eye, and it promotes cyclopia. So more than one option is correct. Okay, so if anyone will try and answer this. Okay, so Indra says number 2 is correct and not very sure about the others. Okay, so anyone else will try to answer this these questions, this particular question? So let us look at the particular question itself. So we are looking at the effect of retinic acid if we take a developing mouse embryo and essentially dip it into a high concentration of retinic acid. What exactly is the result? So this gives rise to retinic acid teratogenesis. So teratogenesis means it is embryonic lethal. What happens is that excess retinic acid causes a shifting of posterior structures to the anterior. So this can be very well explained via the vertebrate column formation. So we have the various bones in the vertebral column which are marked C1, C2, C3, finally going to T1, T2. These are the thoracic segments, this is the cervical segments. But if there is a large excess of RA, we will see that various vertebrate column bones will start forming in the anterior region as well, which should have been in the posterior. So the C7 identity will change to T1, which is posterior to it, and the location of C766 will shift. C5 will not be there. C2 will also shift anteriorly with the absence of C1, and there will be a formation of C0 cervical. In case of RA loss of function, we will see the opposite effect. The anterior structures will start forming posteriorly. So there is a posterior shift. So C1 shifts towards posterior and will replace C2. Similarly, C5 will take the place of C6 and C7, and so on. And this will obviously be embryonic lethal. So this is the action of retinic acid in development. So 2 is the correct answer, shift in posterior structures towards anterior. This also inhibits eye formation, but for our discussion that is not necessary. So this was only one type of one correct answer. So we move on and go to the next question, which is a very fundamental and interesting question, which is a chick's neck has more vertebrae than a mouse, which is obvious because chicks have a longer neck. But what is the molecular basis of it? The options are HOX6 gene is expressed in fewer vertebrae in mouse than in chick. HOX5 is expressed in fewer vertebrae in chick than in mouse. HOX6 is expressed in fewer vertebrae in chick than in mouse. And the expression domain of HOX9 is extended more anteriorly in chick than in mouse. So the options also tell us that this is related to the expression domain of HOX gene, but which HOX gene exactly is it, and how does it tell us that how does it mean that which HOX gene's expression will have will give rise to an elongated neck in chick and not in mouse. So only one type of this is only one correct type of question. So if anyone will try and answer this. Oindira says option 3. So Oindira says it is option 3, that is the HOX6 is expressed in fewer vertebrae in chick than in mouse. So let us look at the solution slide and try and answer this. If anyone else wants to try and look into it. Oh. So let us look into it. So first let me give a brief introduction back to the HOX genes. We have already dealt with HOX genes a lot throughout the course, but I will just go through an important salient point, which is HOX genes are expressed, and HOX genes are the essential body planner genes. Expression of a HOX gene will give rise to a body part in that is responsible for the formation of a body part in that particular region. So there are two important complexes present in Drosophila, which is the antenapedia complex and the bithorax complex. The antenapedia complex, as you can see right here, if the genes antenapedia is expressed in this segment only and will give rise to this segment in Drosophila. So absence of this antenapedia will result in absence of this particular segment in the adult embryo, in the developed embryo as well. So these are very powerful genes. This is the first point. The second is that the position of the HOX genes along the chromosome is very important, as is the position of the HOX gene will be reciprocated in the position of that structure forming in the embryo as well. So say the label forms this particular structure right here. So since this is the anterior structure, label is also present in the anterior, like in the first position, like ahead of all the Hox genes. So there is a co-linearity between the body structures in the embryo and the presence of the genes on the chromosomes themselves. This is a very important point that we have to keep in mind, and we will look into it in the next slide as well. The third point that we have to keep in mind is that in lower organisms such as Drosophila, we have one set of Hox genes, but in higher organisms, especially mammals, we have four sets, and which are Hox A, B, C, and D, and most of them are redundant in activity. So Hox A1, if it is not present, Hox B1 will form the particular structure in the mouse embryo in its place as well. So this is good in one aspect, that if the Hox gene is mutated, the embryo will still develop perfectly, but this spells bad, this has a side effect in developmental biology, as is that if we want to see the mutant or the knockout effect of a particular gene, we have to knock four genes out. That is, say Hox A4, B4, C4, and D4. All of them have to be knocked out in order to understand what exactly the phenotype of the particular gene's knockout is. So knocking out one of them, say Hox B1, will not affect in anything, because all of the other Hox genes will take their place. So these are the 3 points that we get from this particular slide. And next, let us look at into a very important and interesting aspect of the Hox gene. So there is a particular hypothesis that is has been suggested regarding the Hox genes, which is known as the Hox code hypothesis. This suggests that certain combinations of Hox genes specify a particular region of the anterior posterior axis. Just as I said, that there is a collinearity present in the positioning of the Hox genes on the chromosomes, and the particular structure being formed on the final developed embryo. This is what is being suggested with the Hox code, that the position of the region domain of the expression of the Hox gene determines that particular position in the developed embryo. So this is only the Hox code hypothesis, and there are many suggestive facts regarding this. The first important evidence that we can look into is the comparative anatomy. So this is the particular what our question was also referring to. Say in the cheek we have a larger cervical or the larger neck region, and when looked into the particular expression domains, we see that the in chick the HOX5 expression domain is much larger than that in mouse, and HOX5 is what determines the cervical development. So this essentially comes from the Hox code hypothesis, that since the HOX5 is being expressed in a larger region, the cervical region will be larger. So this is the first point regarding this. The second is was suggested via knocking out various Hox genes. So the studies were performed in which, say, Hox10 or the paralogs were all knocked down. So we have Hox10 A, B, A, C, and D all knocked out. And so for context, this is the wild type embryo. So we have thoracic vertebrae up till here. So this is the vertebrate column we are looking at. Then we have the lumbar vertebrae, and then the sacral vertebrae. First, what we do is we knock out the Hox10 paralog, which are responsible for formation of lumbar vertebrae, and we then see that all of them take on thoracic kind of phenotype. So essentially, lumbar is being replaced by the thoracic phenotype, and what that exactly tells us is that if the Hox10 genes are not there, that particular structure will not form in their place. So there is a direct correlation between the gene expression and the particular structure being formed in the developed embryo. This is all. The next point that we see is the formation of the sacral vertebrae as well. Hox11 is responsible for formation of the sacral vertebrae. When Hox11 paralogs are all knocked out, we see that the sacral vertebrae take the are the phenotype seen in the sacral vertebrae is the same as the lumbar ones. So essentially, we can say that the sacral replaced by lumbar, because the Hox11 paralogs are all knocked out. So here also we can see that if the particular Hox gene is not present, the particular structure does not develop. And here we have the evidence other way around. If the Hox gene is present, the particular structure develops for a longer region. So both of these suggestive evidences help in proposing the Hox code hypothesis. So that is also done. Anyone wants to add anything to this? So the correct option is number C. Okay, so let's move on to the next one. We now move into move directly into EvoDevo or evolutionary developmental biology. We have our discussion about the early embryo development is done. We'll now move into the next topic, which is EvoDevo. So the first question that we have from it is, among the options given below, identify the ones that function as modules. So if you remember that we had looked into particular genetic factors that act as modules, and there are other things such as morphogenetic fields that can act as modules, and this help in development, evolutionary development as well. We'll better understand it in the next slide that we'll look into. But the particular question asks us which of the following can act as function as modules. The options are the transcription factor PTEX1, intron that directs formation of tissue specific splice variants, Hedgehog concentration gradient, or limb primordium. This is a more than one option type, correct type of option, because it's a box type. So will anyone try and answer this? Oindrila says it is 1 and 2. The transcription factor PTEX1 and an intron that directs the formation of tissue specific splice variant. Anyone else wants to try and answer this? Okay, so let us look into directly go into an introduction for evolutionary developmental biology and try and understand what exactly we are talking about. So one of the major important things about development evolution is that things evolve. So we know about various evolutionary concepts such as Darwin's concepts of survival of the fittest. Depending on the which phenotype is the best, that will be continued in the next generations. But these, after various experimentation was seen, can only be done via changes that occur during the developmental stage. But since the developmental stage is very crucial to an organism, any changes that occur cannot be very drastic, because that will end up being hampering the development of the adult organism itself. So how does exactly develop these changes of variation in the process of development occur, which should be very stringently controlled? That is exactly what the question is in evolutionary developmental biology. How are the variation changes even if development is such a complex and fine-tuned process without destroying the entire organism? And if the variation arises from mutation in protein coding genes, then the mutant protein will be expressed in all places where the protein is normally expressed. So say if hemoglobin gene was mutated, and then its functions throughout the body will be hampered, and thus it will give rise to it will thus be probably embryonically lethal. But how then? This line of questioning tells us that majority of the variations do not arise from mutations in protein coding genes. So what does a protein coding genes, or there should be particular checkpoints, so that which ensures that even if mutations occur, this will not be dangerous or be hampering the adult organism that develops. So how does it occur? So the answer to the particular question is via modularity and molecular parsimony. So what does modularity mean? Modularity means is that there can be discrete and interacting modules in particular region. So a same parameter can be used in different region and will act differently. So these can include morphogenetic fields, cell lineages, and insect parasegments. So example of DNA region modules can include enhancers. So you have we had already gone through the modularity of enhancer previously, that depending on where the enhancer is present, it so if there is a tissue specific enhancer for a particular tissue, and we take that particular enhancer and put it into the enhancer region of another gene, then that gene will only be expressed in that tissue. So enhancers are modular in that way that we can essentially cut and paste them to different genes, and they will function as is. They do not need any other elements. So this is one of the important points in evolutionary developmental biology, that changes in enhancers can drive cell specific or tissue specific expression for particular genes for which they were not actually present there before. And modular units allow certain parts of the body to change without interfering with function of other parts. So because an enhancer due to a mutation or translocation event between chromosomes, if an enhancer shifts to a gene which was not expressed in say a limb, and this particular gene now gives rise to a novel or a new phenotype in that limb only, then that can give rise to the variation for evolution to work on, since it does not hamper the entire organism. That is what essentially modularity means. So for modularity we have the following examples. So we are looking at the modularity of development in enhancers. So we are looking at the example of a particular type of fish which is known as stickleback. So loss of PTX1 gene expression in pelvic region of freshwater population of 3 spine stickleback. So what happens is that there are two types of stickleback that are present. One is the marine type and one is the freshwater type. The freshwater stickleback does not have any pelvic spines, whereas the marine one does. So scientists wanted to understand on what exactly was responsible for the absence of the spines in the freshwater one. After experimentation, they found that the gene that was responsible for formation of the spines, known as PTX1, is present in both the fish types. But what happens is that the enhancer of PTX1 is mutated so that it does not function properly in the freshwater type, and thus there are no dorsal spines in the freshwater stickleback. So you can see right here the limb enhancer is not properly functional in the freshwater one. But as you can see, the PTX1 is necessary for development in neurons and thymus as well. But those enhancers are functioning perfectly. What this means is that removal of this enhancer just removes the expression formation of pelvic spines and does not affect any other function of the particular gene PTX1. It still acts as performs its particular role in thymus, and it still performs its particular role in the sensory neurons. And this is one of the major proponent for the modularity of enhancer, that if you can remove just one enhancer, it does not affect any others. So if say I put this particular enhancer in the if this enhancer is absent from the freshwater stickleback, and someone took the enhancer from the marine sticklebacks and put it into the enhancer region, we would see the formation of spines in the freshwater ones as well. And this particular experiment was also done in order to show that the enhancers are modular in nature. Thus, the particular option that are correct, among the following options, identify which of the following can act as module. The options are an intron that directs formation of tissue specific splice variant. So we know that enhancer can occur anywhere in the entire genome. They can be upstream to the gene, downstream to the gene, that can be inside the gene as well in the introns. And thus the intron which directs formation of tissue specific splice variant, this is essentially so this does not act as a enhancer itself, it acts as a splice regulator. in which a particular tissue specific variant will exist in various tissues. So example for this is ApoD protein, which in liver gives rise to a particular variant, and in muscles gives rise to another variant because of specific changes present in the intron and splicing. And thus this can also be modular, as if the intron is present somewhere else, it will also function as a module. In case of morphogenetic fields, this includes, as we had already discussed, that morphogenetic fields can also act as modules, and thus Hedgehog concentration gradient will also act as a module. Limb or primordium cannot act as a module, obviously, because in order for the limb to grow, it will still need various other factors which will not be present there. So modules can act independently, but the limb primordium cannot develop independently, and thus obviously is not a module. And transcription factor PTX1 is will not be a module because transcription factors act combinatorially. Then there needs to be various transcription factors present, and thus they are dependent on other transcription factors as well. But enhancers can act instead modular, can be modular instead. So enhancers are modular, independent of everything else, but transcription factors act combinatorially and they are dependent on other transcription factors. So that was that particular question. We move on to the next one. The question is, which of the following is an example of deep homology? The options are: same set of genes specify the nervous system in protostomes and deuterostomes, the ability of mouse PAX6 to induce eye development Drosophila, OTX specifies head formation in both vertebrates and invertebrates, and same Hox ortholog specifies cervical vertebrae in both chick and mouse. So only one option is correct. And which of the following is an example of deep homology? Same set of genes specifying the same thing in protosomes and deuterosomes, PAX6 helping in inducing eye development, OTX specifying head formation, or Hox ortholog specifying cervical vertebrae both in chick and mouse. Which of the following is an example of deep homology? And we will try and answer this. This is only one correct type of question. Okay, so let us go into the particular solution slide. So what deep homology means is that what homology means is that particular genes might be conserved throughout species, such as say Hox genes or PAX6 or PAX type of genes that we have seen that help in eye development. But what deep homology means is that entire pathways made of homologous parts are used for the same function, that they are conserved throughout organisms. And an example for this is the same set of instructions from the nervous system both in protostomes and deuterostomes. So for a Drosophila, we have a plant such as this, in which the signaling molecule DPP and SOG act in opposition, and SOG gives rise to the various structures. SOG gives rise to various ventral structures, whereas DPP gives rise to various dorsal structures. Instead, in deuterosomes such as a mouse, we have a similar event happening, in which cordon gives rise to the dorsal structures and BMP gives rise to the ventral structures. So even though we know that the Drosophila's analog for DPP is BMP, and thus throughout evolution the action of the particular genes are preserved and they act through the same pathways, even though they might not be the same genes, but the pathways themselves are conserved, and that is what is known as deep homology. So this is explained right here, how the highest concentration of SOG gives rise to the midline, and lowering concentration gives rise to VND, IND and various other genes, and similarly highest concentration of cordon gives rise to the midline, lowering concentration gives rise to NKX2, GS6 and MS6, and the opposite happens in case of DPP and BMP4, which gives rise to the dorsal the ventral structure. And we see an the opposite, the exchange of the dorsal and ventral here, because Drosophila, this change has occurred between the protosome to deuterosome, in which the axis gets flipped, but the entire pathway still remains the same. Thus, this also shows that the plan for specifying the animal nervous system may have arisen only once, since the plan is almost similar. Just the genes are also conserved, as well as the pathways to determine the particular structures are also conserved, and thus this is known as gene deep homology, in which entire pathways are homologous instead of individual genes. So the answer to the particular question, which is the following, is an example of deep homology. The answer is same set of genes specify the nervous system in protozoans and deuterosomes. So the ability of PAX6 to induce eye development depends on only one gene, and thus is homology, not deep homology. Same with OTX and Hox orthologs. If the set of genes would have been conserved, then that would be deep homology. So we move on to the last question that we will deal with today, and the question is: bats, but not other mammals, express FGF8 in the interdigital web to prevent apoptosis of web cells. This is an example of which of the following: heterochrony, allometry, heterotypy, or heterotopy. This is the only one type of correct answer, because it is a round type of answer. So would anyone try and answer this? So FGF8 expression to prevent apoptosis of web cells in bats is known as heterochrony, allometry, heterotypy, or heterotopy. Oindrila says it is heterotopy. Anyone else wants to try and answer this? Okay. So we'll go to the solution slide and look into it. So we are discussing, as we are discussing mechanism of evolutionary change. The main mechanisms of evolutionary change are divided into 4 types. These include heterotopy, in which there is a change in location of a gene's expression; heterochrony, in which the time for which a particular gene is expressed is changed; heterometry, in which the amount of the gene being expressed is changed; or a heterotypy, in which the gene itself is changed and another gene is expressed in its place. And these changes give rise to variations on which evolution can act in order to select which phenotype is the best. So if we compare two mammals, the mice and bat, we know that in case of mice, we see that we do not have, the mice as well as us, we do not have anything between our fingers, so there is no interdigital mesenchyme that is present. But in bats, we can see that these are essentially the digits, and we have a webbing that is present in between the digits themselves. So how is this present in bats and not in mice? This is because that bats express the signaling molecule FGF8, which prevents the apoptosis of these cells. So these cells are not removed, and thus they have this webbing that is being formed. So here we can see that in developing sense. So in mice, you can see that in mice and bat, till the third frame, it is almost the same. We have the finger-like projections, the digit primordia, and there is interdigital mesenchyme that is present. But in mice, this interdigital mesenchyme dies out by apoptosis, and we have the finger separating out. But in bat, this does not occur, and the interdigital mesenchyme stays and develops as a web, which the bat uses for flying. And this is because the expression of FGF8 in this particular location. So this is known as heterotopy, which, as we just saw, was change in location of expression. So FGF8 was not being expressed in in the interdigital mesenchyme, but now it is being expressed in the interdigital mesenchyme. That is, its location has changed, and thus this is heterotopy. And thus the bats, but not other mammals, express FGF8, and since the location of FGF8 expression is changed to the web cells, and thus preventing apoptosis of the web cells, this is known as heterotopy, that is change in location. So that was all we had for today. This is the references to the particular slide is Introduction to Developmental Biology by Scott Gilbert, the 9th edition, and this is the YouTube channel link on which all the recorded videos are uploaded. So so if anyone does not have any questions, we can end the session right here. Does anyone have anything to add to the particular session today? Probably not. Okay, then I thank everyone for joining the session today, and I look forward to meeting you next week.